Hello and welcome back to the Biochemistry for Health Sciences channel. Today we'll talk about the three metabolic states, sort of look at the big picture, and then we'll look at the details in future videos. So during or after a meal, we have this state here, what we call postprandial, or after a meal, or absorptive, or fed. So all of these terms have been used to describe this metabolic state. And then while we are asleep or between meals, we have this second state, post-absorptive, also called early starvation or the fasting state. So most of us will go through these two states, the postprandial and the early starvation. Now, if we do not eat for more than one day, so more than 24 hours, then we can enter the third metabolic state, which we'll call either starvation or late starvation state. So let's look at the key or the major metabolic events that happen during these three metabolic states. So first, let's look at the postprandial state, and we'll talk about a normal diet. That means you have eaten normal diet, especially with respect to carbohydrate. So here we're not talking about specialized diets such as the Atkins diet and so on, which will cause a different metabolic sequence of events. But this is just a normal diet with carbohydrates. So during digestion, all your carbohydrate polymers such as your glycogen that you have in your diet or your starch are all broken down to simple monosaccharides such as glucose, galactose, fructose, and so on. We'll just focus on glucose. Other monosaccharides we'll talk about in future videos. So the glucose is absorbed and then it travels through the hepatic portal vein. So portal vein, one which connects one organ to another organ. So it travels through the portal vein and it enters the liver. One of the most important things that happens is this glucose, quite a bit of this glucose, will be used to make glycogen, because this glycogen in the liver, as we shall see later, is the one that will maintain your blood glucose when you are sleeping, when you are in the early starvation state. So once you have eaten something, the body makes sure that this glycogen that was used up in the starvation state, early starvation state, it is replenished, it is remade, so that it can be ready for the next early starvation state. The rest of the glucose will enter the bloodstream, and from the bloodstream it will go to every cell, because there's a lot of glucose. You ate a carbohydrate-rich meal. So the red blood cells will be happy. They can use the glucose, use Main Street and make their ATP. The brain is happy, can make its ATP. So does every other cell, the adipocyte, the fat tissues, as well as your muscle tissues and even your liver. So glucose can be used by every cell, and if there is sufficient glucose, like after a meal, then all the cells will be able to use this glucose for their energy needs. And remember, this is the only time when the blood glucose is very high. Blood glucose that is very high will stimulate insulin production by the beta pancreatic cells. So this is a time when your insulin levels are very high after a meal. So although glycogen is found in most cells, most of your glycogen in your body is found in the liver as well as the muscles. So this synthesis of glycogen from glucose, what we call glycogenesis, will be predominant in the liver as well as the muscles. Now humans can convert glucose into fatty acids and then use the fatty acids to make triglycerides as well as other lipids. So humans can do that. We can make glucose into fatty acids. We cannot do the other way around. We cannot take fatty acids and make glucose, but we can make glucose into fatty acids. So that's another important pathway that happens after a meal, and that is fatty acid synthesis. Now, adipocytes will take these fatty acids and make triglycerides, which they will store in here. These triglyceride droplets, these are very big droplets stored in adipose tissue, in adipocytes. And these triglycerides, we'll see later, become very important for energy production, especially during the starvation state. Now, the liver is also a very important organ as far as synthesis of lipids. So it can take excess glucose and convert it to fatty acids, as well as other lipids, cholesterol and triglycerides. Now, the liver, although it makes a lot of lipids, it does not store the lipids. Okay, it takes these lipids, makes them into lipoprotein particles, what we call very low density lipoprotein, and it secretes this VLDL out into the bloodstream so that they can be used by other cells. Okay, so very important point. The liver makes lipids, but it does not store lipids. Okay, if you have too much lipids in the liver, that is abnormal. We actually call that fatty liver. Fatty liver, if there's too much lipids in the liver, that is not normal, that is abnormal. And we'll talk about this in future videos. Dietary proteins are broken down into amino acids in digestion, and then once again they travel through the portal vein to the liver, and in the liver these amino acids, some of these amino acids are converted into various proteins. For example, albumin. Albumin is very important, it has many functions, and we'll talk about this in future videos. The liver is where albumin is made. The liver also makes a lot of other proteins, such as your blood clotting proteins. The rest of the amino acids will enter the bloodstream, and they will go to every cell. So, for example, in the brain, a lot of these amino acids are converted to neurotransmitters. Most of your amino acids will end up as proteins in the skeletal muscles. Okay, so as far as protein storage is concerned, as far as how much protein we have in the body, most of your proteins will be in the skeletal muscles. Now, dietary lipids, after digestion, you will get the building blocks, such as fatty acids as well as cholesterol. Now, unlike glucose or the amino acids, these building blocks, lipid building blocks, do not go to the liver. Instead, they are assembled into lipoprotein particles called chylomicrons, and they enter the lymph vessels and eventually into the bloodstream. So these lipids will appear in the bloodstream as chylomicrons. So these chylomicrons will travel through all these cells, all the various cells, various tissues, and in the tissues the fatty acids will be removed from the chylomicrons, and these chylomicrons become smaller and form what we call chylomicron remnants. And then these chylomicron remnants will be taken up by the liver, where they'll be broken down into the building blocks once again, such as fatty acids and cholesterol. Now, dietary nucleic acids are broken down into nucleosides, and then they are reconverted into nucleotides, and most of the purine nucleotides from what we know today are used by your GI system. The pyrimidine nucleotides, there is evidence that pyrimidine nucleotides may be sent to the liver. So it looks like we humans do not depend on nucleotides that we get from the diet. We prefer to make them ourselves. Okay, so what about early starvation or the fasting state? Probably the best example is, let's say, 2 or 3 o'clock in the morning when you are fast asleep. Now recall from an earlier video that red blood cells can only get their energy, their ATP, from glucose. The brain gets 80% of its ATP from glucose. So it's very, very important to have an optimum level of glucose in your bloodstream. Since there is no glucose coming from the GI system, from your diet, so the question is, where is this glucose in your bloodstream coming from that is so essential to your red blood cells as well as to your brain? And the answer is liver glycogen. So liver glycogen is broken down by this process called glycogenolysis. Lysis means breakdown, so breakdown of glycogen. So glycogen is broken down into glucose. Glucose is exported into the bloodstream, and this glucose will be used predominantly by your red blood cells and your brain. Note that although muscles have a lot of glycogen, their glycogen is not used to maintain blood glucose. That does not happen. Now the brain, for example, needs 120 grams of glucose a day. The rest of the body needs 40 grams. So that's about 160 grams roughly of glucose we need every day. Your liver glycogen can give you about 200 grams of glucose. So there is enough liver glycogen to maintain your blood glucose for about 1 day. Okay, 24 hours, 1 day. Now, it's important to note that in starvation state, our insulin levels are very, very low, and what rises is glucagon. So glucagon that is secreted by the alpha pancreatic cells starts rising as we enter from the fat state into the starvation, the early starvation state. So insulin levels go down, glucagon levels go up. Now there is another process that also slowly starts happening as we go from the fat state into the early starvation state. And this process is called gluconeogenesis. Neo means new, genesis is synthesis, gluco is glucose. So this is synthesis of new glucose, not glycogen, not the glucose from glycogen, but synthesis of totally new glucose. So the liver takes building blocks of glucose, two most important being pyruvate and oxaloacetate, and converts it into glucose. And then this glucose can be exported out into the bloodstream and be used, especially once again by the red blood cells and the brain, to make sure they have enough glucose for their ATP needs. So where do these building blocks such as pyruvate and oxaloacetate come from? The answer is they come from proteins, and since most of your proteins are in your skeletal muscles, so what happens is these proteins in the skeletal muscles are broken down into amino acids, the 20 amino acids, but then these amino acids are converted into two major types of amino acids, glutamine and alanine, and these two travel into your bloodstream and enter the liver, and in the liver these amino acids are converted into the building blocks of glucose so that the liver can make glucose via gluconeogenesis. Now since these amino acids have nitrogen, so when we remove the nitrogen comes out, is eventually converted into urea, and this urea eventually will be excreted by the kidneys. Now notice, although the adipocytes have triglycerides, which can be broken down into fatty acids, and these fatty acids can be sent to the liver, but notice that these fatty acids cannot give us the building blocks, pyruvate and oxaloacetate, and that's why humans cannot convert fatty acids into glucose. Okay, so we'll talk about this problem here, very important problem in future videos. So although fatty acids which come mostly from these triglycerides that are stored in the adipocytes. So fatty acids are released, enter the various organs. Now in the liver, although the fatty acids don't give us the building blocks, but what the fatty acids gives the liver is ATP, is energy, because gluconeogenesis is a synthesis pathway, and synthesis always requires energy. So fatty acids will give the energy part. So while the building blocks come from muscle proteins, the energy for gluconeogenesis will come from fatty acids. So in starvation, in early starvation, so you can see the glucose is used only by red blood cells and by the brain, while the fatty acids are used for energy generation in all the other cells, adipocytes, muscle cells, liver cells, and so on. Okay, so while the rest of the body is using fatty acids, the glucose is being reserved for your red blood cells and for your brain. Now there is a little bit of fatty acids here, you can see, that is used by the brain. So it's been reported that the brain can get about 20% of its energy from fatty acids, but more than that it becomes toxic. So the brain still relies 80% on glucose for its ATP needs. Okay, so you get up in the morning from your early starvation state, and then you have your breakfast and you go back into your postprandial state. So humans basically go between those two metabolic states, postprandial as well as early starvation. But we do have another state, and this is called the late starvation state. So we're going to divide late starvation states into two phases. Let's talk about phase 1 first. So the first important point to note is what differentiates late starvation from early starvation, and that is there is no more glycogen present in the liver. Okay, so there is no more glycogen. So now your blood glucose that is so essential to your red blood cells and to your brain, what maintains your blood glucose now is gluconeogenesis, the synthesis of new glucose from simple building blocks such as pyruvate and oxaloacetate. So what we saw in early starvation becomes more dramatic in late starvation. More and more proteins in the muscles are being broken down into amino acids, releasing which are converted to glutamine and alanine. These glutamine and alanine are then transported to the liver, and there they are converted into the building blocks for glucose and then made into glucose via gluconeogenesis. The nitrogen of these amino acids is excreted as urea. And at the same time, a lot of triglycerides, which are mostly in your adipocytes, are broken down to fatty acids, and these fatty acids are used for energy generation, ATP generation in your adipocytes, in your liver, in your muscles, and in all the other tissues. So while your brain and your red blood cells are using glucose, everyone else is using fatty acids. So the production of ATP from fatty acids is a very important process that happens it begins in early starvation and then becomes very predominant in late starvation. Again, this process is called beta oxidation. We'll talk about beta oxidation in the future video. So beta oxidation is how we generate ATP from fatty acids. Okay, how we generate ATP from fatty acids, beta oxidation. So gluconeogenesis, fatty acid, triglyceride breakdown, as well as beta oxidation become very predominant in early starvation and then becomes more predominant in late starvation. Okay, so this is phase 1, the early part of late starvation. Now, what happens if you still do not eat anything? Okay, what happens if you get into the later part, phase 2 of late starvation? As we get into the later part of late starvation, remember our proteins, our muscle proteins are being broken down to amino acids, because these amino acids will form the building blocks for glucose, and that glucose is essential to keep us alive because your red blood cells in your brain needs that glucose. But the problem with breaking down your muscle proteins is that it is going to make you very weak, right? Your muscles are breaking down. You cannot walk, and this is a program that is supposed to help you find food, and to find food you need your muscles. Okay, so for survival you do need to walk around and find food to get out of the starvation state, but the problem is your muscle proteins are being broken down, so that's not good. So to slow down this protein breakdown in your muscles, what the liver does, it not only uses the fatty acids for generating ATP, it also uses the breakdown product of fatty acids, acetyl-CoA, and converts the acetyl-CoA into ketone bodies. These are water-soluble small molecules. These enter the bloodstream, and here's the important point. Ketone bodies can be used by the brain to get ATP. Ketone bodies cannot be used by your red blood cells, but they can be used by the brain to get ATP. So this means now the brain will rely less on glucose. Okay, so it relies less on glucose. The red blood cells, unfortunately, still can only use glucose. They cannot use ketone bodies. So protein breakdown will still happen, but it will slow down, right? It will slow down because now we only need the glucose to feed mostly the red blood cells and not the brain, because now the brain is going to be using ketone bodies to generate this ATP. Ketone bodies can also be used by other tissues, such as your heart and your skeletal muscles and so on, to generate ATP. So this is a summary of what are the key events that happen postprandial, during after a meal, early starvation, that is between meals or while you're sleeping, and then late starvation when you do not eat for more than 24 hours. Of course, there is the metabolism of other very important biomolecules that are present in the body. For example, your membrane lipids, nucleotides, as well as chemicals such as heme, that is very important for your cytochromes and myoglobin and hemoglobin. And we'll talk about all of these in future videos. So please keep this big picture in mind as we go through the various metabolic pathways, such as your beta oxidation and your fatty acid synthesis and so on in the next few videos. Always keep this in mind because it helps you see the big picture. And again, very important for healthcare professionals to see the big picture so that we can connect biochemistry with other classes such as physiology, anatomy, pharmacology, pathology, and so on. So that is all for now. Thank you very much, as always, for watching these videos. Hope they will help you as you travel through your journey to becoming a healthcare professional. Goodbye for now and have a wonderful day.